students of St. Peter's College, Colombo, Colombo branch. Today I am going to talk about Unit 7 of Grade 13 Biology. We are going to discuss today some topics related to DNA sequencing, DNA isolation, and enzymes related to gene technology. If we go to DNA sequencing, we define DNA sequencing as a process to determine the precise order of nitrogenous bases in a DNA molecule. It means there are nitrogenous bases in a DNA molecule and we are going to detect the order and we are going to type the order. If we recall the DNA molecule, it is made with 4 bases: adenine, guanine, cytosine and thymine. And these bases are arranged in a DNA molecule due to hydrogen bonds, and these 2 DNA strands are antiparallel. The nitrogenous bases are written in an order to a particular gene. And this sequence is linearly arranged. And this sequence is unique to DNA molecule and its genes as well as non-coding DNA sequences. If you recall a DNA molecule, these are the structures of the nitrogenous bases, and this is the way it has arranged in an antiparallel manner, and these are the hydrogen bonds, how they are arranged chemically, and this DNA molecule is converted to RNA, actually mRNA, in transcription and in translation to proteins. So, the DNA sequencer is written in amino acid sequence language in protein synthesis. DNA sequence is done by an automated machine known as a DNA sequencer. And this is a DNA sequencing machine, and these are a series of DNA sequence machines. And this technique was introduced in 1977. After the introduction, this technique was improved greatly. And at the beginning, some manual techniques were used, such as 2D chromatography, and it was time consuming. So when you feed DNA to a DNA sequencer, the output is an electropherogram. This is an electropherogram and it shows peaks to all the 4 bases, and these are the color code they have used, and these color codes are depending on the machine, and it is a plot of DNA fragment typically obtained by genotyping via automated DNA sequencer. Inside a sequencer, what happens? We feed the DNA like in agarose-gel electrophoresis to a sample buffer, and in these tubes gels are running, and there's a fluorescence detector to detect each nitrogenous base. So there's a laser to detect in the detector, and after the detection these peaks are formed, and this is how it is running, and this is how it is typed. This is a real electropherogram of a particular gene, and you can see how it comes as strips of plots, and under the plot the nitrogenous bases are there. When you avoid these peaks, the bases are written like this. This may be having coding sequences as well as non-coding sequences. And this technique has greatly improved, as I told. In 2003, Human Genome Project was initiated. It means the entire human genome was sequenced, all the possibilities of genes of humans. And the technique used were Sanger sequencing method or chain termination method, and it is known as the first generation sequencing. And it was time consuming because only small fragments of DNA was able to sequence, and several countries were contributed in this effort, and actually it took 15 years to complete. But now it is greatly improved. Now there are modern methods of DNA sequencing. It is known as next generation sequencing or second generation sequencing. Simply we call next gen sequencing. And the most modern method is third generation sequencing using nowadays. And this is the novel technique. And the great improvement in this is the time required for sequencing is greatly reduced. It took 15 years to complete the Human Genome Project, but we can tell our own DNA in several hours in a low cost. For that, we are using only a small amount of our tissue samples, such as hair, blood, or any other tissue you have. So there are many applications in DNA sequencing. It is used in molecular biology and evolutionary biology, to classification as well as phylogenetic relationships, and medicine to great extent, and forensics and metagenomics, and also the DNA fingerprinting. In molecular biology, it is used to understand the functions of DNA. In molecular biology, we are studying biological molecules. DNA is a specific biological molecule. To understand the function, we are using DNA sequence because we can type it easily. And also, certain domains of DNA can specify the functions of a protein, whether protein has what functions and its order. This is the DNA sequence, and this is the amino acid sequence, and this is a polypeptide, it means a protein. By studying them, we can deduce whether it is a transmembrane protein having specific function, or it is a DNA binding protein, or an enzyme. So, also presence of multiple copies of DNA we can deduce after typing it using DNA sequencing. And also, it is easy to get the amino acid sequence also using the DNA sequencing, because now we know which codons are coding for a particular amino acid. In evolutionary biology, this is used to a greater extent. We know DNA is passed from generation to generation, and we can know the similarities and differences of each individuals in its own species, as well as their relationships to other species. So DNA sequencing is important to study them, because we can sequence it and we can see whether that particular nucleotide order is there in other species too. So actually, though it is we are discussing it manually, this is done using vast number of programs, softwares. And using these sequences, we can draw phylogenetic trees to show the relationships, and also from ancient human samples such as mummies or bodies buried in ice or fossilized samples, we can study about ancient humans as well as ancient organisms, and we can get a complete study of them and how they are related and how they are migrated to different areas of the world. All these things we can study using DNA sequencing. This is a phylogenetic tree of fungi, and using different sequences we have fed to the computer, to a particular software, and we can draw this phylogenetic tree and to show their relationship. We can see ascomycetes and basidiomycetes are related here, and how each are related to each other also we can get through a phylogenetic tree. In medicine also we are using DNA sequencing a lot. We can understand a healthy person, whether it is a carrier or not. This is the control sample, this is the patient sample. We can see the tweet here and also the nucleotide order here. Other than this, a T has come. So now the amino acid sequence has changed into valine. It is glycine, so it causes a disease. So we can understand whether a person is healthy or not, especially in their genetical diseases. And we can diagnose cancer also in the similar manner, and we can follow the drugs given to cancer using DNA sequencing. We can actually see whether DNA carried in the blood of the patient, whether those mutant alleles are increasing or decreasing, and through the analysis of placenta of a fetus, we can identify whether the fetus is having a genetical disorder, and we can early diagnose whether a disease is present in the fetus, and we can get treatments or necessary advices after studying these DNA sequences. So in metagenomics also we are learning about DNA sequencing. We are using a lot, and it is metagenomics is a science in which DNA present in an environment is extracted as community DNA and studying this sample as a whole. It means microorganisms we study as a whole. Earlier we used culturing methods in this case, but most of the microorganisms are unculturable. So when we sequence it, it is easy to study. We can identify the previously present ones as well as living species, and we can use it in ecology, epidemiology, and various other fields related to this microbiology. And we are studying the total microorganisms in metagenomics. DNA sequencing in applications of DNA fingerprinting. DNA fingerprinting is used in forensics. In our DNA, in our chromosomes, there are some non-coding sequences. Coding sequences gives a particular product always. It produces a particular protein. But in some areas there are non-coding sequences, and scientists have understood there are some repeating non-coding sequences. These are short sequences. 2 to 6 base pairs are repeating here. All of must have these within our DNA. Actually, between the genes, these are located. Those are known as short tandem repeats. Short tandem repeats are usually denoted by STRs. These STRs are analyzed in DNA fingerprinting. So usually these STRs are variable in individual. Here individual A, B, C, D, and E. Here 4 repeats are there. In AA is the STR and it is repeating 4 times here. Here 5 times. In C it is 6 times and in D it is 7 times and in E it is 8 times repeating. So it is unique to an individual. So it can be used in identification in forensics as well as paternity testing. So this is how we are using DNA sequencing in forensics. This is suspect A and suspect B. And suspect C is there. So except for identical twins, usually these STRs are not unique to each other. So using that principle, we are using these STRs in criminal identification. Here the crime scene DNA, it is a DNA sample obtained at the crime scene and it is showing these STRs and it is matching with suspect B, not suspect A all the time but exactly with suspect B. So we can use it for criminal identification. With C also one is matching but others are not matching, so it is matching exactly with suspect B. So these STRs are first sequenced and run in an agar cell so we can compare them and deduce whether suspect is or not. So paternity testing also used when the paternity of a child is in doubt. So for that also STRs are first sequenced and then run in an agarose gel. This is a gel of these STRs. This is the mother's STRs and this is the child's. Some have come from the mother. We don't have a doubt in that, but father is a doubt. So male 1 and male 2 are suspects here and we are matching. These STRs are related to male 1 or 2. First two STRs are present in male 1. All the 4 has not come but these 2 are here. But these 2 STRs have come from mother and here again the child has this STR related to male 1. But in the first one actually it may have come from male 2 also. Male 2 also have the same. But this STR is present in male 1 only. So like that we are comparing all the STRs and we are getting the highest percentage of STR match to test whether the paternity is related to which male. And also to find out the biological grandparents especially in European countries, the relationships are distinct and sometimes after a long time when somebody wants to clarify the biological parents, this method is used. And this is my question on DNA sequencing. Write an essay on DNA sequencing and its applications. And I'm going to the next section now. It is isolation or extraction of DNA. It is also a technique in molecular biology. Usually, uh, we have to isolate the DNA sequences from the total gene, which means all the genes in a particular cell or an individual. So, the donor cells can be animal, plant, or microbial cells. We can get the total genome from that. So, all the DNA we are going to isolate or separate. So, DNA molecules are very long. When we are extracting DNA, some damages may occur. DNA molecules may break. We have to minimize it in this process, otherwise we can't use it in our activities. So, this is a cell suspension, and finally the DNA is extracted here using a procedure, but it is depending whether it is a plant or an animal or a fungi or any other bacterial cell. And even viral DNA we can extract. For different organisms we have different procedures. So it has used in many applications. Extraction of DNA is important in other applications such as DNA structure studying in chemistry and DNA protein interactions, especially in molecular biology, and the DNA hybridizations and the DNA sequencing. It is a molecular biology technique, and PCR is also a molecular biology technique, and uh, genetic studies and gene cloning to produce transgenic organisms. So, this is the common procedure of DNA isolation. So, first the DNA is inside the cells, so the cells should be disrupted, or it is known as cell lysis, and DNA is an enzyme in the cell, so we have to inhibit it because DNA is usually cuts or breaks the DNA, and DNA is associated with nucleoproteins, so we have to dissociate it, and now all the other organelles and everything is there in the suspension, so those are contaminants, those should be removed, and then finally the DNA is precipitated to a pure, as a pure culture. So the first step is homogenization or disruption or lysis of cells. In this we are using a lot of mechanical methods. So this is some sound waves are used here, and yes, you can use high pressure, or some equipment can be used here like a blender or a homogenizer, and cell is broken into, the membranes are broken and the contents are out. So we are using mechanical methods as well as chemical methods. So these are the mechanical methods, and in chemical methods we are using cell lysis solution. It is actually having lysozyme enzyme which are in the lysosomes, which causes cell lysis. So the second step, inhibition of DNase. DNase means a DNA degrading enzyme. Deoxyribonuclease is the chemical name of that. Now the cell is broken. Now the DNA is in touch with this enzyme. So we have to avoid that. So we have to inhibit that enzyme. So for inhibition, some chemicals are used. Those are known as chelating agents. And these are some common chelating agents. EDTA is the chemical structure, and this is tris-EDTA, and there are some more. And the third step is dissociation of nucleoprotein complexes. We know DNA is bound with histone proteins, so we have to get pure DNA. So in that case also, we are using some chemicals. The most commonly used chemicals are SDS, phenol, and proteolytic enzymes of proteases. So these chemicals also breaks the amino acid sequences in the nuclear proteins, and this is SDS and phenol doing the same thing. We know proteolytic enzymes are used in our body also in digestion. There what happens? Proteolytic enzymes break the peptide bond and amino acid molecules are released. Anyway, now the enzyme can't be activated anymore. So we are deactivating this. So now we want to extract DNA. So all the other materials, the chemicals we added, as well as cell debris and even the cell organelles are now contaminated. So those contaminations should be removed. In that also we are using some chemical methods. Some applications it is necessary, and all the contaminants are removed using some chemicals such as phenol, and when phenol is mixed, cell is having water a lot, so we can get two phases. In the aqueous phase, we can get the nucleic acids, DNA and RNA both, but we want to extract DNA. So, in the next step, what we do is we are going to actually extract DNA. We are using cold ethanol in this, and DNA is really dissolved in the buffer here, but we have to remove RNA also. We are adding RNA, DNA is free RNA is used. It is ribonuclease is used to remove RNA. Now we are getting only here the ethanol is used, and we are dissolving it. When we dissolve it comes in this way, and DNA can be seen in a test tube or the centrifuge tube as a small cloud here. So there are some enzymes also which we are using in gene technology. So we are going to see what are these enzymes and their actions. And the enzymes we discuss here are restriction endonucleases, DNA ligases, DNA polymerases, and reverse transcriptases. So we are going to see each of the enzymes and their functions. So restriction endonucleases. Endonucleases means they are cutting the DNA. In some procedures which we are using, such as cloning, we have to cut DNA in particular precise sites. In that case, we are using these restriction endonucleases. We are simply known as restriction enzymes. They cut the DNA in specific sites, and these sites are known as restriction sites or cleavage sites. And in cloning, actually, two DNA fragments should be cut, because one is in the usual plasmid DNA and also another DNA which should be cloned. So both are cut, and it should be ligated. For cutting, we are using restriction endonucleases. It recognizes specific DNA sequences and cut at nearest site or restriction sites. And most commonly used restriction endonuclease is EcoR1, and it is an enzyme obtained from E. coli. The second enzyme which we are using is DNA ligase. It is in DNA replication mechanism also. DNA ligase is ligating. It means it actually seals the cut ends. Here, ligation, it is the cut end or sticky end. It is using DNA ligation. It is ligated. And here what happens actually, it forms a ligation, means it forms a phosphodiester bond. And most common DNA ligase which are used in DNA cloning is obtained by T4 DNA bacteriophage. And most of these enzymes are obtained from bacteria or microorganisms. DNA polymerases, it is actually adding complementary deoxyribonucleotides to the template strand. Here, DNA polymerase here, they are adding, and we can see this is extending. So, and it also does proofreading, and we know DNA polymerase into 3 to 5 direction, and new strand is made into 5 to 3 direction. So, it is actually copying the DNA, and it is used in PCR as well as DNA sequencing. And when we are using DNA polymerase in those procedures, we have to use Taq DNA polymerase, which is heat stable, and it is obtained by thermophilic bacteria. And the next enzyme is reverse transcriptase, and reverse transcriptase is doing the reverse of transcription. It means using an mRNA sequence, it forms a DNA sequence, and this DNA is known as complementary DNA. So it is the replication DNA polymerase is used here, and here reverse transcription means again a DNA is formed, and we know in RNA other than thymine uracil is there, so it is replaced with thymine when you make DNA. This DNA is known as cDNA. This is the reverse of transcription, so that's why the enzyme is known as reverse transcription. And cDNA is very important in some molecular biology techniques. So this is the end of the lesson, and thank you for listening. And these are the questions to explain briefly the enzymes related to gene technology and also the procedure of DNA isolation. Thank you.